This is slide 51, and it's our last topic for chapter 21B. So there are four ways that antibodies fight against antigens. What the antibody does is it binds to the antigen, and many antibodies can bind to a single antigen. And what happens is they form this antigen-antibody complex. The four ways that antibodies can defend against antigens is neutralization, agglutination, which we've seen with the wrong blood type, being transfused, precipitation, and complement fixation. So I'm just going to show you a little illustration here that shows you once the antibody binds to an antigen, it can neutralize the antigen just because many of these antibodies bound to that antigen, it can't really affect other cells. It becomes not dangerous anymore or not pathogenic anymore. It can cause agglutination, as you see with these blood cells clotting. It can cause precipitation. When antibodies bind to antigens, sometimes they can cause that antigen to precipitate out of solution. And that just means, like if it was dissolved in solution, now it's not dissolved anymore. And that makes it easier for phagocytes to engulf. And then you can also have complement activation, which once an antibody binds to an antigen, that can activate the complement system. And remember, we talked about how complement can form holes in the surface of a microorganism and enhance phagocytosis and different things like that. So antibodies also are going to enhance phagocytosis and inflammation and can lead to the lysis of harmful cells. So we're going to move on to part C, 21C. I'm going to open up that set of slides. And this will be our last set for the immune system. All right, section 6, which is the cellular immune response, of course, which is regulated or controlled by T cells. T cells provide defense against intracellular antigens. That means antigens that are inside of our cells. So this would be cells infected with viruses or bacteria, also cancerous or abnormal cells, and foreign or transplanted cells. Some T cells directly kill these types of cells, these types of harmful cells, and others release chemicals that regulate the immune response. You do need to make sure you know the difference between CD4 cells and CD8. These are just two different types or populations of T cells. So if they're CD4 cells, they tend to become helper T cells, or T subscript H. Helper T cells can activate B cells. Helper T cells activate other T cells and macrophages. And helper T cells basically direct the adaptive immune response. So CD4 cells usually become helper T cells. CD8 cells usually become cytotoxic T cells, which means that these are T cells that are capable of killing cells that contain foreign antigens. So here are your major types of T cells. They can be CD4 or they can be CD8. If they're CD4, they usually will become a helper T cell. And if they're CD8, they will become a cytotoxic T cell. Okay. Now, T cells respond only to processed fragments of antigens displayed on the surface of cells by MHC proteins. So they only respond to antigen presentation. Antigen presentation by an APC, antigen presenting cell, is vital for the activation of naive T cells. That means T cells that haven't been activated, and the normal functioning of effector T cells. So there are two classes of MHC proteins. There's class 1, and all cells display class 1 proteins except red blood cells. And then there's class 2, which are displayed by antigen-presenting cells. Remember, that can be dendritic cells, macrophages, and B cells. Class 1 MHC proteins are crucial for CD8 cell activation. So what do we need to remember so far about CD8 T cells? CD8 cells become cytotoxic T cells, and they're activated by class 1 MHC. And then for CD4 cells become helper T cells, and they're activated by class 2. And this is just a table comparing and contrasting class 1 and class 2 MHC proteins. All cells have class 1, only APCs have class 2 MHC proteins. All right, and then this section talks about MHC, major histocompatibility restriction. CD4 and CD8 cells have different requirements for MHC protein that present antigens to them. CD4 cells that become helper T cells are restricted to binding to only class 2 MHC proteins on APC surfaces. We already said that. It's just summarizing. CD8 cells that become cytotoxic T cells are restricted to binding only class 1 MHC proteins found on every cell surface, including APC surfaces. So that's important to remember, is class 1 can be found on every cell surface, including an APC, whereas class 2 is only going to be found on antigen-presenting cells. And I like this down here. It says if the cell that's displaying the class 1 protein is an antigen-presenting cell, that is telling the immune system, I belong to self, that I have captured a foreign invader. This is what it looks like, the antigen. Kill any cell that displays it. If the cell is not an antigen-presenting cell, it's saying, I belong to self but have been invaded, like by a virus, or become cancerous. Kill me. All right, T cells can be activated only when the antigen is presented to them. Activation is a two-step process involving antigen binding to the T cell and co-stimulation. Both of these processes occur on the surface of the same antigen-presenting cell, and both are required for the clonal selection of a T cell, so for the T cell to start dividing and mounting an attack. All right, in antigen binding, the T cell antigen receptors, we call those TCRs, that would be a receptor on the surface of the T cell. These TCRs bind to the antigen MHC complex on the APC surface. So the T cell receptor must perform double recognition by recognizing both the MHC and the antigen. Other T cell surface proteins are involved in T cell activation. For example, CD4 and CD8 help maintain coupling during antigen recognition. Then there's co-stimulation. Complete T cell activation requires the T cell to also bind to one or more co-stimulatory signals on the surface of the APC. So this two-step process ensures that there's not like T cell activation when we don't want T cells to be activated. We want our immune system to work, but we don't want our immune system to go crazy when there's no reason. Then there's proliferation and differentiation, which is when the T cells are activated and they begin to divide and they differentiate to become memory T cells or helper T cells or cytotoxic T cells. Activated T cells are a hazard because they produce large amounts of cytokines, which induce the inflammatory response. And this could result in hyperplasia or cancer if not cleared from the system. And hyperplasia is just when an organ enlarges, like the kidney enlarges or the liver enlarges. So this shows you the process. It shows you that here's your bacterium that's causing your infection, enters your tissues, encounters a dendritic cell. That dendritic cell engulfs the bacterium and displays the antigen attached to a class 2 MHC protein. Okay? And then there's also, in addition to that, there's a co-stimulatory molecule here. Let's blow this up, because I'm having trouble seeing it, and I'm actually using a laptop. So you have the APC, which is displaying the antigen and also the co-stimulatory molecule. Then it's going to encounter a T cell, which has a T cell receptor that's going to recognize the class 2 MHC and the antigen and the co-stimulatory molecule. And in addition to that, a CD4 protein is going to attach and ensure that the process is even, the fit is even more tight, I guess you could say. Okay? So that, all of that together, the CD4 protein, the MHC, the antigen, and the co-stimulatory molecule bound to the T cell receptor, all of that is going to ensure that the T cells begin to divide and clone. And some are going to form more helper T cells, and some are going to form memory T cells in this case. All right, and so then the cytokines are released by T cells. They are the chemical messengers of the immune system. They include interferons and interleukins. These are some different ones listed and what they do. They enhance the immune response, enhance inflammation. Helper T cells activate both branches of the adaptive immune response, the humoral and the cellular immunity. So they activate other T cells as well as B cells. Okay? Without helper T cells, the immune system doesn't work. Okay? So once the helper T cells are primed by antigen presentation, by the antigen presenting cells presenting the antigen, the helper T cells then help activate B cells and activate other T cells. They cause the proliferation of other T and B cells, and they secrete cytokines. Without the helper T cells, there is no immune response, and that is the reason why HIV is is terrible for your immune system. It shuts your immune system down because HIV, the HIV virus, HIV, human immunodeficiency virus that causes AIDS, attacks helper T cells. So that's how it shuts the immune system down. The activation of B cells occurs when helper T cells interact directly with B cells, displaying antigen fragments bound to MHC2 receptors. So when the B cell is an APC, basically. And it just shows you here how that works. You have your helper T cell bound to an MHC class 2 antigen complex, and then you have a CD4 protein sort of making that binding process even more secure. And then you have the release of these cytokines. Interleukins are being released in this picture, and all of that ensures the efficient binding of the helper T cell to the B cell, and it activates that B cell. Now, we're going to move on and do CD8 cells because we've been discussing CD4 T cells, so now CD8 T cells, which become cytotoxic T cells. CD8 cells require helper T cells to become activated into and become like able to destroy and kill harmful cells. The activation of CD8 cells cause dendritic cells to express co-stimulatory molecules required for CD8 cell activation. So CD8 cells require helper T cells, and they require co-stimulatory molecules, basically. All right, so you have the interleukins here that are being released are the co-stimulatory molecules. You have the helper T cell, and this is our CD4 cell. Now, if you look down here at the CD8 T cell, it's got to bind to the MHC class 1 and the antigen, the CD8 protein, and the co-stimulatory molecule, and then also the interleukins that are being released. So all of that together ensures proper activation of the CD8 cell. And we'll stop here.